Scientist and author Lyle Watson once said, "If the brain were so simple that we could understand it, we were really so simple we couldn't." And this quote nicely describes the enormous scientific challenge that we face in trying to understand the human brain and how it functions, how it relates to our cognitive abilities. And even though over the last decades we have made enormous progress in brain imaging methods, finding new insights into how the brain functions, how the architecture of the brain is laid out, and how that relates to complex cognitive abilities like perception, memory, spatial orienting, and many, many more, I would argue that we basically have no idea how the brain actually works in our natural environments. And this is because brain imaging methods investigate human brain dynamics in participants that are stretched out supine in a scanner, that are sitting in dark experimental rooms, not allowed to move at all. This is because the sensors are either too heavy to follow participants' movements, like in an MR scanner of several tons, or because the movement induces movement-related artifacts that distort the feeble signal of interest that originates in the brain. So my contention here is that if we really want to understand the human brain and how it naturally functions, we have to leave the standard lab setup. We have to go and measure brain activity in actively behaving humans. As for now, we record millions of bits of information regarding brain activity, and we compare that with 1 bit of behavior, a button press at the end of the trial. That seems to be like a fundamental mismatch. And does that reflect how our brain works in our natural environment? I have serious doubts. The brain evolved and is a very highly energy-consuming organ, and it evolved basically to survive, to increase the probability of survival of the human species. It is not sufficient to develop color vision to be able to sense the health level or emotional state of other people. It is to select the appropriate action plan if an ill-tempered, orange-faced primate comes up. It is not sufficient to remember that a red fruit has higher sugar levels than a green fruit, or that the seriously weird-smelling food makes you sick. It is to remember where to find the sugar-laden fruit again, or to inhibit eating food that is too old. Perception is for action. And memory is not retrospective, but prospective. It did not evolve to reminisce about the past. It evolved to be better prepared for action in the future. So let me give you 2 examples why behavior or action is so important for cognition and why it is important for brain dynamics. I'm going to show you a video of a single cell recording in a rat. So in animal studies, we can record cell activity in actively moving animals, and that shows a tight relationship of movement, behavior, and brain dynamics. What you're going to see is a rat running around an elevated track, and what you are about to hear is the firing of single cells deep in the rat's hippocampus, deep in the brain of that animal, that fire whenever the rat is in a specific place. That's why these cells are called place cells. So here you can see the rat, and now the rat is moving, and you hear the firing of different place cells. And certain cells fire when the rat is in a specific place, other cells will fire when the rat is in a different place. With that kind of information in other cells, like grid cells and heading-sensitive cells, the rodent is able to compute a perfect metric representation of our environment. We can do the same. We have the same cell types in our brain. So the point here is that the activity of these cells are obviously influenced by active behavior, by movement through space. The second example that you're going to see is activity, again, single-cell activity of cells in the brain of a fruit fly. And the activity was measured in 2 different conditions. One condition was the fruit fly was resting, not moving at all. And the second condition, the fruit fly was airborne. The trick here is that the fruit fly is glued to a stick. It does not really move in space. It flies, but does not move. In both conditions, the fruit fly perceives the exact identical visual input. And if you look at certain region of the brain that you can see here on the left-hand side, the activity, that's the brain, the upper part of that animal, and here you see activity of certain cells in that brain in an area that is not directly related to motor output. That's not the area that controls flying, but does other computational things, and the moment the animal is airborne, you can see that the activity changes dramatically, basically indicating that changes in behavioral state, even though you're not moving, you see the same things, completely lead to a change in the brain dynamic state. More importantly, the movement itself will lead to sensory output that will be used to reach an action goal. Sensors in your muscle, in your vestibular system, in your ear that provide the basis for balance, for movement and orientation changes in space and how you react to that with your body in upright posture, basically provide you with all the information that you need to compute spatial relationships without visual input. You can easily test it. I'm sorry, but in the first talk I have to ask you to please stand up. Seriously. Okay, so look around you. Try to fixate like one of the windows. Look at one certain object that you see in this room. And now face me again, please. And close your eyes. Keep your eyes closed until I tell you to open them again. And now slowly turn on the spot to your right. Slowly until I say stop. Turn around on the spot. Turn around. Full body rotation. Turn. More. And stop. And now, without opening your eyes, please turn back to face the object that you just fixated before. If you've done so, open your eyes. And you see that you should be more or less accurately be able to face that spatial location again. Thank you very much. You can sit down. So basically you were able to update a position in space that you couldn't see. During active rotation, you changed your position and orientation in space, and still you had a complete and fine, accurate representation of where that object location was without visual input. That is a demonstration of how important our bodily senses are for cognition and for other higher processes too. So, in standard or established brain imaging studies, we do not have the means to investigate moving participants. As I said, the scanners are too heavy, or we are afraid of movement-related distortions of the signal. So how do we measure activity in actively moving participants? 10 years ago, I started to work with Scott Makeig from the University of California in San Diego. And we developed a new mobile brain-body imaging. We called it a mobile brain-body imaging, or short MOBI. Dan Ferris joined us from the University of Michigan, now Florida, to conduct a series of experiments where we investigated participants walking, standing, and running in their environment, interacting with their environment to better understand how the brain dynamics works. So mobile brain-body imaging basically overcomes restrictions of established brain imaging methods by using portable mobile EEG systems. 25 years ago, this wouldn't have been possible. This is a 32-channel EEG system that is mobile completely, transferring data wirelessly to your handy, to whatever kind of mobile device, using Bluetooth or whatever kind of protocol. This is technology that developed lately and that allows us to record EEG, electrical activity of the brain, originating in the brain, in actively behaving participants. However, if you want to understand how movement impacts brain activity, then we have to synchronize the EEG with the movement. So we have to synchronize EEG with motion capture. In addition, we can synchronize other data streams like head-mounted virtual reality, peripheral physiology, whatever you would like to synchronize. It took some years to overcome the technical challenges of synchronizing wireless data streams, megabyte, with limited bandwidth, without delays in the different data streams. But now we have two labs, one in San Diego and one here in Berlin, that is fantastic. It's the Berlin Mobile Brain Body Imaging Lab, part here of the TU Berlin. That is a dedicated research facility to mobile brain body imaging experiments. And what would that look like? So here you see a participant in an experiment, and she basically does exactly the same what I just asked you to do, keeping up orientation while turning on the spot. And we're interested in trying to see which brain areas represent that kind of vestibular and proprioceptive information that is used to reorient towards a certain heading or orientation. And what you can see is a 160-channel cap of actively amplified EEG on the neck and over the head to measure the electrical activity of the brain and the neck muscles. In addition, that's synchronized to the motion capture. You can see the red LEDs that allow us to track position and orientation changes of participants, head, torso, full body motion capture. And that is synchronized to the head-mounted virtual reality system here that allows us to control visual input. And as Margaret Wilson nicely stated it, cognitive processes are deeply rooted in the body's interaction with the world. I'm going to show you an example of an experiment that clearly demonstrates that brain dynamics are also deeply influenced by active behavior and that changes cognition. One more word to the data. Because participants are moving and they produce movement-related artifacts that heavily contaminate the signal of interest. So we need to use data-driven analysis approaches to dissociate brain from non-brain activity, like neck muscle activity, eye movements, or mechanical-induced artifacts due to the movement, cable sway and electrode movements. So we need advanced computational methods to dissociate all these different contributions to the sensor-level data that we record. So I'm going to show you an experiment, and here we basically exported a very established experiment that we know in psychology for decades now, where participants have to respond to a target that appears on the screen. And we compared two conditions here. We compared that participants in one condition had to simply press a button whenever a moving sphere changed the color to the target color. And in the second condition, we asked participants to actively point and intercept the trajectory of that moving sphere. So here you can see the setup, and you can see a participant basically showing motivated action, not simply sitting in front of a screen and just pressing a button, but this is the active pointing condition here. And what you can see is that the sphere moves, it changes its color, and then participants are asked to respond to that by touching the moving sphere. So this kind of activity is a natural activity that we observe in our daily life nearly every other minute. If you would like to drink some water using the glass in front of you, what you have to do, you have to grasp the glass. Before that, you have to encode the location of the glass with respect to your body. That location of that object is encoded based on a retinotopic coordinate system. That is a coordinate system that is centered in your eyes. However, if you want to grasp it, you have to align that retinotopic coordinate system with the coordinate system of your hand because you want to grasp that glass. That is connected to your arm, that is centered in your shoulder, providing yet another coordinate system that has to be aligned with the visual coordinate system to allow controlled movement. This is heavy computation. We don't notice that at all. It's a simple task for us, seemingly, but the computational demands underlying that are quite great. If you compare that and make the glass moving, like in this experiment, the object is moving, the computational load increases. Parietal cortex, an area in your brain somewhere here, is one of the cortex areas that, among other functions, provides the basis for the integration of different coordinate systems. It is not involved in execution of motor processes, though. It's not a motor area. It's a multimodal integration area. Here you can see the activity of different participants localized to the brain, and three different perspectives that you can see here. And this is the parietal cortex, and we were interested how this brain region contributes to the sensor level data that we record. And what you can see here in this picture is on the left-hand side, the activity time-locked to the onset of the color change in the condition on the left-hand side where participants simply had to press a button. And you can see that a target that appears and that is response relevant leads to significant activity in the parietal cortex in the time range that we would expect from traditional brain imaging studies. If you look at the pointing condition, however, you see, importantly, a significantly increased activity, even though that area is not involved in executing the motor movement. So this shows that continuous computation and alignment of different coordinate systems in a dynamically changing world is simply computationally more expensive. The data that we see in MOBI experiments are not comparable to the data that we know from established brain imaging studies because we restrict all these aspects of natural behavior, of natural interaction with a complex and dynamically changing environment. We are far from understanding how the brain actually functions. Where do we go from here? With MOVI, if we are able to understand how active behavior influences cognition and brain dynamics, and how brain dynamics during active behavior change motor aspects and cognitive processes, we will have a better insight into one important aspect of how our human brain works and functions. With that, we can investigate the impact of movement on perception. Why and how does our perception of the environment changes when we execute certain motor programs? What is the relationship of movement to spatial orienting abilities and the underlying brain dynamics or memory in general? If we know better how the brain controls motor behavior, we can develop better diagnostic and therapeutic approaches to treat movement disorders like Parkinsonism, stroke patients, rehabilitation approaches. We open up a new universe of new approaches, including central nervous system activity, to improve cognition and movement. And most important for me, we do gain a better understanding of how a naturally embedded brain functions in an active human interacting with a dynamically changing world. Thank you.